Good afternoon. On behalf of the Global Interdependence Center's Board of Directors, welcome to today's executive briefing. I'm Bill Kennedy, GIC's Vice Chair of Programs, and I will serve as your moderator. Today, we will examine virus mutations, specifically how the genetic structure of coronavirus changes and how therapeutic and vaccine technologies can adapt to meet the challenge. As a reminder, this event is being recorded. The slides from our presenters and the replay will be available on the GIC website starting tomorrow. I'm pleased to introduce our two speakers. Dr. Priya Nori is an Associate Professor of Medicine and Orthopedic Surgery at the Albert Einstein College of Medicine. She serves as the Medical Director of the Antimicrobial Stewardship Program and Outpatient Parenteral Antibiotic Therapy Program at Montefiore Health System, where she also serves as Program Director of Infectious Disease Fellowship Program. She's an alumna of the Johns Hopkins University and Albert Einstein College of Medicine. Dr. Thomas Unnasch is Distinguished USF Health Professor at the College of Public Health at the University of South Florida. His research has focused on vector-borne diseases and human filarial infections. He is the director of USF's only high containment laboratory for infectious diseases and was named a USF Distinguished University Professor in 2020. Dr. Unnasch received his AB from Rutgers University and Ph.D. from Massachusetts Institute of Technology. Dr. Nori, Dr. Unnasch, welcome, and thank you for joining us. So Dr. Unnasch, you will be first. Thank you very much. You will be first, and the floor is yours, and we look forward to your presentation. Over to you, sir.
Okay, I'm waiting for the slides to appear. Am I running the slides, or?
Now, we'll have the slides coming up.
Okay, great. So what I'm going to do today, I think Priya and I are going to be tag teaming, and I'm going to try and give a primer on virus mutations and how they occur and how they might be selected for, with particular emphasis on SARS-CoV-2, the causative agent for COVID-19. Can I have the next slide, please? So what is COVID-2? COVID-2 is one of the coronaviruses, and the reason that they were named coronaviruses is pretty obvious, I think, from the left-hand picture on the slide here. This is an electron microscope image of a coronavirus particle, and you can see it kind of looks like a coronet, a medieval crown. Corona is Latin for crown, and they were named as viruses that look like crowns, essentially. On the right hand is a 3D reconstruction of what the virus really looks like. If you actually could drill down and be the same size as the virus and look at it, you can see it's kind of a spherical particle, but it's coated on the top with these large protuberances that are sticking out that are these spikes that form the little ridges around the crown of the coronavirus that's shown in the electron micrograph on the left-hand slide. Can I have the next slide, please? So coronaviruses are actually very common infections in animals. They're extremely common in bats, which is where SARS-CoV-2 is originally thought to have originated. There are like 2,000 or 3,000 documented different types of coronaviruses that infect bats overall. But within the human system, there's really only 4 or 7 coronaviruses that have been identified so far. 4 of these circulate quite widely and just cause the common cold, the sniffles that you get in the wintertime, very similar to a rhinovirus infection. 3 of them, though, can cause serious infections in humans, and these are all coronaviruses that have made the jump recently from animal hosts into human hosts. The first is severe acute respiratory syndrome, or the SARS-1 virus, which circulated around the world in the early part of this century, the 21st century, began again in China, probably as a result of an infection in an live animal market through a civet cat, it's believed. Middle Eastern respiratory syndrome, or MERS, is the second coronavirus that's out there. It's a virus that's found primarily in camels and can make the jump from camels to humans. Occasionally, though, there's been not a large amount of human-to-human transmission. it has a very high case fatality rate of around 50%. So it's a really unpleasant infection to get. Found primarily in countries in the Middle East and very commonly in Saudi Arabia, where people tend to have camels and buy camels and trade camels quite a lot. And then, of course, we have SARS-CoV-2, which is the second severe acute respiratory syndrome virus, which is the cause of COVID-19 or the 2019 coronavirus disease. Can I have the next slide, please? So this kind of gives you the structure of the coronavirus. Up on top there is a picture of how the genome is structured. The genome is structured into a relatively large number of open reading frames, which encode for proteins. There are several of these that are labeled the ORFs that are there that are actually that code for polyproteins, which are long strands of amino acids that are then cut up subsequently to being translated and turned into individual proteins. Most of these are involved in the non-structural part of the virus, the mechanism that allows the virus to replicate itself and assemble in the cell. On the surface of the virus particle, there are really about 5 or 6 different proteins. We talked about the spike glycoprotein originally. There is also a membrane glycoprotein and an envelope protein. And then inside the virus, the RNA gets packaged inside this little capsule, and it's wrapped around another protein known as the nucleocapsid protein or the N protein. Can I have the next slide, please? But almost all of the action that you're going to hear about today and all the action that we've heard about for vaccine development really is focused on the spike protein. And the spike protein has got an inordinate amount of attention as a result. This is for several reasons, some of which are biochemical and many of which are immunological. First, the spike protein encodes the receptor binding domain of the virus, which is the key that allows the virus to bind to and invade our cells and therefore infect our cells. The receptor binding binds to an enzyme that's on the surface of the cell known as the angiotensin converting enzyme, which is an enzyme that's involved in regulating your blood pressure. The ACE2 receptor is for short. The spike protein is also the major target of the immune response to the virus. Almost all the antibodies that neutralize the virus that you get out of natural infections appear to target portions of the spike protein. So not only is it important in terms of the receptor binding of the virus, but it's also important immunologically because it seems to be where most of the action for developing a neutralizing response to the virus is located. As I said before, the protein's about 1,273 amino acids in length. It's about 180,000 Daltons to 200,000 Daltons in molecular weight. That's the difference is because after the protein is translated, it's decorated by the cell with a lot of sugar residues or glycoprotein. It's a big glycoprotein. And so these polysaccharide residues add to the molecular weight. And there are varying amounts of glycosylation that occurs that causes differences in the overall molecular weight that you see. Could I have the next slide, please? So if we look at how viruses evolve, they evolve according to the old Darwinian principle of natural selection. And this can really be divided into 3 sorts of steps. The first is just mutation, which are random changes in the sequence of the genome that can occur. And these occur all the time, as we'll talk about. But you can occasionally get a mutation that occurs that gives the virus some sort of a selective advantage. And then you have natural selection that will occur that will basically choose these newly more fit viral particles, and they will take over the virus population eventually and replace the less fit versions that are out there as well. We also can have what's called founder effects, which are basically an effect when you have a small virus population, let's say from a single individual who may move to another country or another area and begin to infect people there. It's a very small population, of course, it's only in a single individual. So whatever random viral particles that individual is carrying are the ones that get introduced into the new country. And you have what's a so-called founder effect, resulting in a genetic bottleneck that will select for certain types of viruses. And this is actually very useful for tracking viral spread around the globe. Can I have the next slide, please? So RNA viruses, which are, as the coronavirus are, tend to be really subject to relatively high amounts of mutation. And the reason for this is that the RNA polymerases that replicate these genomes are pretty error prone. So SARS-CoV-2, for example, has a mutation rate of about 1 in a million per site in the genome per cycle of replication. This is about 30-fold lower than influenza, which has a mutation rate of about 3 times 10 to the minus 5, or 1 in 300,000 mutations per site per cycle. Influenza is about 30 times more mutagenic, or makes 30 times more new viral mutations per cycle than SARS-CoV-2 does. The reason for this is that SARS-CoV-2 actually has a little bit of a error correcting proofreading activity in its polymerases, and so it's able to correct any mutations that occur, which is better for the virus to maintain itself, but at the same time slows the mutation rate, which is good for us. I think we'll hear about that a little bit later. Replication mutations and mistakes in the replication process result in one change at a time. So you don't get multiple changes, it's one change at a time. We can also get recombination though. If you have two different types of virus, strains of virus that infect the cell at the same time, the replication RNA polymerase can jump from one to another and make the front end of one genome from one viral isolate together with the back end of another viral isolate. And this can introduce large numbers of mutations simultaneously and cause a reassortment that gives you a relatively quick amount of mutational reassortment and shuffling at a single step. Can I have the next slide, please? So for the mutations that we're talking about, the mutations that are made by the RNA polymerase, there are really three different types of mutations that you can have. Transitions are the easiest ones to make, which are a purine replacing a purine. A purine nucleoside has two rings that are interlocked, one 6-component ring and one 5-component ring. And these were adenine to guanine, and that therefore, since they're pretty similar from their structure chemically, you can get A to G changes or G to A changes fairly easily during the replication process. You can also have pyrimidines, which are just a single 6-component structure ring, and that would be cytosine to thymine or thymine to cytosine. Transition, again, it's a similar structure, so the mistake can be pretty easily made by the polymerase. Transversions, on the other hand, where you have a pyrimidine that replaces a purine or a purine that places a pyrimidine, they're a little less common because obviously a two-ring structure is very different than a one-ring structure, so the polymerase is a little less likely to make that mistake. And finally, you can have deletions or insertions that can occur when the polymerase slips and stutters and can introduce extra base pairs or delete certain base pairs. Can I have the next slide, please? And how do these actually make a change? Well, getting back to your basic molecular biology, there are 64 different codons that are out there that can be encoded in a standard genome. These are groups of 3 nucleotides that specify a particular amino acid. Since you have 4 nucleotides and that you can have A, G, C, or T, and you need 3 nucleotides to make a codon, that's 3 to the 4th, or 64 different codons that are possible that you can make with the different combinations of the 4 nucleotides. Out of these 64 codons, 3 of them just tell the ribosome to stop, that they've come to the end of a protein, which will leave us 61 codons to code for our amino acids. But in nature, we only really have 20 amino acids that are out there normally in proteins that are made, which means that there's a lot of redundancy in the genetic code. For example, the amino acid leucine is coded for by 6 different codons, which are shown here: CCT, CTC, so on and so forth. And you'll notice that it's CT and any nucleotide at the end. So if you have a mutation that occurs in the 3rd position of that codon, it's never going to change the codon, what it codes for, from a leucine to anything else. It'll always be leucine to leucine. So this redundancy helps reducing the amount of mutations that can occur during the amplification process or during the construction of the proteins, which is good for the organism in general, but slows again, the importance of the mutations. So can I have the next slide, please? So we really have different types of mutations that are out there. We can have silent mutations, like I talked about before. If you have a mutation in that third position in the leucine codon, it'll always still code for a leucine. So it doesn't really make any difference structurally or functionally to the virus or to any other organism that has the mutation. On the other hand, you can have missense mutations. These are mutations which result in a change in the amino acid that's coded for in the RNA structure. And these are the ones that are really important, that will really make a difference. And then we finally have mutations of concern. Okay, and these are identified by mutations that make a change, that make a substantial change in the protein, that changes the behavior of the protein. And in our case, it's mostly the spike protein that we're really interested in. And you'll see a lot of this mentioned in the literature that's out there, and how they're identified in these mutations. They're enabled by the amino acid position where it occurs in the spike protein, by the identity of the residue that was there originally, and then by the new mutant residue. And we always use these as coded for by single amino acid designations that the code for. For example, a really important mutation that I think you'll hear about a little bit later is the E484K mutation. And this is a mutation that changes the 484th amino acid in the spike protein from a glutamic acid to a lysine. So the structures are shown below. Glutamic acid is shown over there on the left, and the lysine is shown on the right. You can see they look fairly similar except for the very end of the side chain. But the glutamic acid actually has an acidic residue up there, and at neutral pH that develops a negative charge. And the lysine has a amine residue at the end, which under neutral pH has a positive charge. So what you're doing there is you're changing a negative charge to a positive charge. And if you have an antibody that recognizes the negative charge and it now has a positive charge, it's almost like a magnet going from attracting to repelling, and that antibody is not going to bind that protein nearly as well as it did previously if you have a change from the negative charge to the positive charge. Can I have the next slide, please? So once we have these mutations, then we can have different types of selection pressure that will develop on them, that will select these variants to actually overgrow and start to take over. Some of them that you can think about are host range. That's how the virus made the jump originally from bats to humans. Virulence, the virus can either cause a greater amount of disease or a lesser amount of disease. There could be pressures that move either way. Infectivity, if the virus changes its spike protein to bind more tightly to the receptor, it can increase its infectivity and it'll be selected for. Of course, we'll have escape mutants. Once we have a large population of people who are immune, there's going to be pressure on that virus to avoid our immune response. And so we're going to get so-called escape mutants that will begin to develop, that will begin to avoid the immune response that we have to natural infections and vaccinations. And finally, eventually, you can imagine if we develop decent drugs against this virus, we'll have drug resistance that can develop as well, which we'll also have to deal with. And with that, I think I'll turn it over to Priya.
That's great, Thomas. Thank you very much for sharing your thoughtful remarks, really laying the groundwork for Priya's presentation. But please stick around. We're going to ask you to come back for the Q&A in just a moment.
I'll be here. I'm staying on.
Okay, great. So Dr. Nori, the virtual floor is yours.
Yes, thank you. And thank you, Dr. Unnasch, for such a high-level setup. I hope now to take it further into the clinical realm and in terms of implications for what this means for our path forward. Next slide, please. Okay. So just to review and add on to what Dr. Unnasch had so beautifully explained, there are several variants that are pretty concerning for those of us in public health, on the clinical side of things, and really for every sector of society. These are the ones to watch, basically. And taking what Dr. Unnasch explained and taking that a step further, these lineages, these variants, contain multiple mutations that actually change the way that the virus behaves and leads to some kind of selection advantage, meaning that it's a ultimately more fit virus. And so not only do we have one version of that, but we have at least these three fairly dominant versions and potentially several more as we continue to follow this pandemic. And so the ones to keep in mind are the UK variant, which is technically called the B.1.1.7. And recall that this had its origins in the United Kingdom, and by their very high level and widespread sequencing that they do very nicely in the United Kingdom, this was traced back to actually September, the fall. And this contributed largely to the exponential growth of the virus between the fall, peaking in January, and then has subsequently declined to some extent due to vaccinations. But why this is scary is a couple of reasons. One, that now this thing is in multiple countries, over 90 countries, and almost all of the states within the United States. Because we now have several months of experience with this virus, we actually know how it behaves. So it does a couple of concerning things, which is that it's doubling at a rate of every 10 days in the United States. So whatever facts that we cite, whatever statistics we cite 2 weeks ago really don't even apply anymore. You can kind of multiply that by X number. This is on track to become the predominating variant in the coming weeks if we don't do all of the other public health measures that we know work. And because we've seen how this can actually play out in the clinical arena, some reports, studies from the National Health Service in the United Kingdom show that it's up to 35% more deadly and between 30 and 50% more transmissible. The other big ones, the important ones to watch, are kind of similar in that they share certain scary mutations. One is the South African variant, which has its origins there, was tracked, initially identified in December. And the really scary thing here is that the mutation, which Dr. Unnasch mentioned, these quote unquote EEK mutation, we call it that because it's somewhat scary. This makes it rather resistant to lots of the therapeutics we have in our arsenal. And I will get more into that shortly, but this mutation is also shared by the Brazil variant and also by this emerging New York City variant, which is certainly concerning to me as someone practicing in New York City in the Bronx. Next slide, please. Okay, so again, there's lots of mutations. Each one of these variants contains potentially dozens of mutations, but the important ones, as you learned previously, are the ones that affect certain critical functions. Receptor binding to the human host cells, cleavage, and then potentially downstream functions of things like replication. And so these two here that I've circled are the ones to watch because not only do they affect therapeutics, but potentially vaccines and also how quickly we can get these the current wave of the pandemic under control. Next slide, please. And so I had mentioned just a few seconds ago that this newly branded New York City variant, which was identified by a laboratory out of Columbia University, seems to have features similar to the Brazil and the South African variants in that it may be particularly resistant to some of the tools in our toolkit, including monoclonal antibodies, convalescent plasma, potentially certain vaccines. Next slide, please. Okay, so what is ultimately from today, what are the take-home points about these variants? If you can memorize the numbers, that's fantastic, but you really don't need to. But understand that they're all pretty similar. They have a similar group of mutations that have come to light because they confer an advantage to the virus, a fitness advantage. They lead to stronger binding to the host cells. They lead to evasion of host immunity, including exogenous antibody therapies, and they potentially may make certain vaccines less effective, especially if we don't work quickly to update those vaccines and test them against existing variants and anything that we have in the repository of mutations. Some may be more deadly, the UK variant, but that has yet to be seen with these other variants emerging from either South Africa or Brazil or New York City. But these will likely go up before they come down. And so it's truly a race between vaccines and variants. Next slide, please. Okay. So what I do in the hospital, my role at Montefiore Medical Center is that part of what I do for COVID is that I help to oversee a monoclonal antibody treatment program. And so this is a program that lots of hospitals basically stood up very quickly with emergency use authorized monoclonal antibodies made by either Regeneron or Eli Lilly to treat the community, to treat outpatients who are early in their disease course, where the viral levels are really spiking very high on the order of millions of copies. And the science shows that the earlier you can treat these types of patients with neutralizing antibodies to block the binding of the virus, the better they're going to do. And they're going to avoid hospitalization, oxygen needs, intubation, and all of the very bad sequelae that we've come to associate with monoclonal antibodies. However, next slide, please. You can click twice. There's an animation actually to show that as these variants take a stronger footing and are more prevalent in the United States, the shelf life for these antibodies is going to be very limited. Right now, they're doing a fairly nice job at preventing severe outcomes in patients who are at particularly high risk, but there will come a time, just like with antibiotic resistance, but with viruses, it actually happens more quickly. There will come a time where these therapeutics are potentially no longer valid, and they're sort of yesterday's news unless we can quickly update these things, these drugs. What does this mean in terms of vaccines? So let's just review quickly, what are we working with in terms of vaccines? So the two earliest vaccines to obtain EUA from the federal government are the two mRNA vaccines. And so these are made by Pfizer and Moderna, and this is what most healthcare workers got. This is what essential workers, nursing home residents, and lots of the first and second tier of society have gotten these vaccines because these are truly what was authorized first for widespread use. However, very exciting news. Recently, the Janssen vaccine was also authorized, and this is because after extensive, highly rigorous review, this vaccine has also shown to be extremely effective, especially in preventing the worst outcomes. Next slide, please. Okay, what does the story with variants being kind of right, maybe a few steps ahead of therapeutics, maybe one step ahead of vaccines, what does this all mean for us in terms of our goals to reopen society safely, to resume doing the things we enjoy, to invigorate the economy, and to get back to some, or to arrive at some new normal state? What does all of this mean? How we roll out our vaccines and our strategy, public health strategy, is very critical here, because it seems that deviation from the protocol in terms of how these vaccines, especially the 2-dose vaccines, were studied either 3 weeks apart or 4 weeks apart, it seems that deviation from that could potentially be risky, because an individual is only partially protected after their first dose. And so if they were to become infected with a virus that was of one of these variant lineages, this may not be recognized, or there might not be a robust enough immune response from the first dose of vaccine to prevent severe illness in that individual. However, that being said, if you look at current curves now in the United Kingdom, it looks like the worst of their curve, their epidemiology curve, was in the early part of January. And things have really come down nicely and leveled off, though not at a very low level. They've leveled off at a steady plateau. And they did adopt a strategy of extending the interval between the first and second doses, because the thinking at the time was that if you at least partially immunize lots of the population, perhaps that's advantageous to fully immunizing only a smaller percentage of society. So something they're doing is certainly working. And I think part of what they did extremely well, in addition to having a really good handle on sequencing on large scale national databases of sequences, is that their vaccine rollout was also one of probably the most effective in the world, also similar to Israel. One piece of good news is that vaccines do a lot more actually than just create antibodies or just prompt your body to create antibodies. There's a lot more to it than that. And so while antibody levels may be, let's say, or the efficacy of the neutralizing property of the antibodies may be less than initially hoped, lots more is happening in the immune system, in our lymph nodes, in our spleens, in all these different places that are responsible for a robust immune response. I'll get into that a little bit more in a few minutes. And so these antibodies created in response to vaccination are not just for that very narrow region that is mutated based on these viral mutations in these variants, but it's more broad than that. These antibodies are broadly neutralizing in that they target the entire spike region and not just a couple of base pairs or a couple of amino acid sequences that may be off. So while efficacy drops somewhat in the face of variants, if the end game is to prevent death, hospitalization, and severe disease, all of the vaccines that we have available through the FDA EUA are extremely robust. And so that is truly the good news here, that less people will die. Less people will ultimately shed the virus and infect others. And ultimately, this is a gigantic breakthrough. So boosters will be likely, undeniably, I think, especially if these variants are in heavy enough circulation, especially in the United States. The FDA already has a plan to urge vaccine companies to update their available vaccines. And so groups like Pfizer, Moderna have already submitted the paperwork for the initial steps to studying these vaccines. Luckily, enormous clinical trials are no longer needed for this process. The initial vaccine trials had more than 40,000 people, about maybe 20,000 and 20 to 30,000 in each arm. The FDA stated that this is not going to be needed. It'll be more of a fast track process, so we won't need to wait another 12 to 18 months to know what these booster shots look like. Next slide, please. Okay, what about this issue of reinfection and infections that are occurring in a small number post somebody being fully immunized? So the definition of full immunity is somebody being 2 weeks from either their second dose of mRNA vaccine or 2 weeks out from the one and only dose of the Johnson & Johnson vaccine. So while a couple of months ago, before these variants were sort of predominating, the thinking was that reinfection truly doesn't happen outside of a really minor handful of cases. Those hosts are usually immune compromised in some way, and this was not really thought to be a big issue. However, it's becoming clear that reinfection due to variant strains is entirely possible. The best example of that is in Manaus, Brazil. Hope I said that right, where in the initial wave, up to 2/3 of their population was infected. They had a very high hospitalization and death rate, unfortunately. And the seropositivity in this city in the Amazon basin was very high. And the thinking was that it's nearly close to herd immunity. However, fast forward a couple months later, people who had been infected and had documented immunity were starting to become reinfected. And this was linked to the P1 variant really leading the pack in terms of new infections. And this particular variant has something like 17 important mutations which affect the way the spike protein binds to the host cell and potentially evades existing immunity. We have seen on the clinical side of things, we certainly have seen infections occur after people are considered fully immunized. The good news is, though, that this is an extremely small number compared to all of the people that we've vaccinated so far in New York City. It's happening, but ultimately the percentage of that will be extremely small, in my opinion. If you read the studies from Israel, it really seems that this is a rare occurrence, and that truly what this is attributed to is the fact that you're very highly protected after your 2 vaccine doses. And also, it seems to have a very positive effect on that person's, not only their susceptibility to infection, but also their shedding of virus, their infectivity to those around them. So I think that we're starting to get a hint of this now from reports coming from different parts of the world, from the UK, from Israel. I think this will be a very favorable outcome in terms of how infectious are fully immunized people are they able to infect those around them? Okay, so this is what I would put in the category of cautious optimism. Next slide, please. Okay, so this is what I alluded to before, which is that while the ability of the antibodies created in response to vaccination may be somewhat less than we'd hoped in the face of infection against these new variants, it's certainly not cause for giving up. These vaccines have by no means failed in any way. And in fact, they're a huge, resounding success. Because again, just to emphasize, the end game is to prevent people from dying. And then along the way, we're going to prevent people from infecting each other. And we're going to really hopefully arrive at a place where the virus is under much better control. If those are the goals, all the vaccines we have available are highly effective. And part of this is because, again, there's multiple arms of the immune system, only one of which is the creation of antibodies. There are cytotoxic T cell responses, there's helper T cell responses, there's B cell responses. These cells also prime other types of immune cells. They communicate with each other through complex chemical signals to make sure that once there's a reintroduction of virus or that individual is exposed again, the immune system is ready. And there's multiple infantries. The first one being the antibodies, and then the second line being, multiple other NK cells, cytotoxic T cells. There's lots and lots happening that is ultimately very favorable and very protective. And they were able to prove this by testing volunteers who had been fully immunized or who had been infected with variants. They tested their sera against variants, and they also tested their lymphocytes, and they found a high degree of protection. So this is very good news. Keep going, please. Okay. One thing we can't ignore, we can't deny the contribution of non-pharmacologic interventions. So these are the things that we know are extremely effective. They're probably as effective as vaccination in preventing illness if we adhere to them in an extremely strict way. So masks, social distancing, physical barriers, all of these quote unquote public health measures, they don't give a lick about variants. They don't know which person is infected with the variant and which person is infected with a quote unquote wild type virus. They work regardless. They're highly effective regardless. There have been a few updates to the guidance about this. In the beginning, it was, surgical masks. Now it's potentially double masking or one really good mask like a KN95 or an N95. But regardless, these things have held up beautifully, okay? These are ideal. They're low cost. They're sustaining interventions. They're easy to implement, and they're incredibly effective. The other major leg of this is testing. Part of why certain countries are definitely leading the pack in terms of pandemic control is that they have extremely robust testing infrastructure that not only includes PCR-based testing, but also rapid antigen point-of-care testing that is utilized in multiple points of access. So what this looks like to me in an ideal state is that something like 70 or 80% of society is immunized, basically all eligible adults. Plus, we offer testing at multiple points of care, not only in your doctor's office or an urgent care center, but heck, a take-home test from CVS or one of the retail pharmacies, in school, in airports, in vending machines, in restaurants. And this, a negative test result combined with documentation of full immunization from a vaccine is essentially our passport. These are two very important legs of us being able to get back to some new normal state. Next slide, please. And I think that's almost towards the end of my talk, but what is the United States doing about all this business with variants? While our ability to sequence is not yet as robust as we need it to be, they're working hard at this. The CDC has partnered with local public health laboratories to sequence a bunch of viral strains from different hospital systems throughout the country on a regular basis so they can at least know what's in the system, where are the hotspots, et cetera. And they are partnering with private labs like Abbott to ramp this up on a huge scale. So something like thousands of specimens per week that they're hoping to sequence, develop a sequence library, and to understand and to track this throughout the country and throughout the world, and then also use this to refine therapeutics, use this to refine vaccine doses, and apply this towards booster shots that may be needed in the future. Next slide, please. Okay, so I think this is the final. So what does this all mean? Is coronavirus here to stay? Can we expect some degree of endemic virus? Is this going to revisit us every year? The answer is quite possibly. I think so. I think what our new normal is going to look like some kind of low-grade level of virus that may have hotspots every so often. And so if our vaccines are truly effective in preventing death and hospitalization, which they are, if they're truly effective at preventing disease transmission between individuals, which is looking like they are, and if we can vaccinate enough individuals to really reach this herd immunity threshold, then I'm optimistic that we can reach a very optimistic new normal, as long as we keep lots of the public health measures that we know work and really really invest a lot in terms of a more robust, multifaceted testing infrastructure in this country. So that's it for me. Sorry I went a little bit over, but really happy to be here and take questions at this point.
Oh, thank you, Dr. Nori. That was fantastic. So we'll bring Dr. Unnasch back on. And now we're going to move into audience Q&A. So I'd just like to remind our attendees, if you'd like to ask a question, just go to the dashboard, go to the questions tab. You can type in your question and that will put it into the queue. So while we're waiting for some other questions to come in, one question that has been asked: what mitigation advice do you offer immunocompromised vaccinated persons? Dr. Nori, do you want to take that one?
Sure, I can certainly start. So the concern about immune compromised individuals. So these are folks who are on immune suppressing therapies, who've received chemotherapy for a cancer diagnosis, who have had an organ transplant or something. The concern is that they won't mount as robust an immune response to the vaccines, both in terms of antibodies and some of these other immune cells that I mentioned. However, we've not really proven that, because that type of subject was not within these clinical trials. We're making assumptions about what we've seen in terms of other vaccines and other diseases. And so these individuals should absolutely still be prioritized for vaccination. And they should be prioritized for repeat doses of vaccination and booster shots. But until things are truly to a very comfortable, sustained, controlled level, even a fully vaccinated but immune compromised individual ought to continue to really mask aggressively, especially in public spaces. Think about where you absolutely need to travel to and where you may not, and think about issues like crowd control, and there's lots of things, that mitigation strategies that still can be utilized if we're not as confident in one's response to a vaccination.
Okay, great, thank you. So let me just, Dr. Unnasch, are you still with us?
Yes, I am.
Okay, great. I'll put this one to you. The participant is asking, why is the J&J vaccine, or is the J vaccine, which is using a viral vectored approach, versus the Moderna using an mRNA approach, is it better for your body in the long run?
I like to say that it's a lot like a difference between a red M&M and a brown M&M and a green M&M. The coatings on the outside are all a little bit different, but what's inside is almost pretty much the same on the three vaccines. There is some concern about the J&J vaccine. I think that I've heard about that. If we have to go to repeated injections, people may start to develop an immune response to the viral carrier itself, and that will reduce the efficacy of those shots. Whereas the Moderna and the Pfizer vaccines are pretty much a totally synthetic construct. It's unlikely that any sort of immune response is going to, off-target immune response is going to develop to those. But I think all of them are basically almost exactly the same. They're raising antibodies against the same spike protein. And as I'd like to say, if it were me, I would just get the one that was available to me first.
Yeah.
Dr. Nori, you agree?
Sure, absolutely. I wanted to add to that I heard, Dr. Unnasch, maybe listen to this podcast too. I'm a really huge fan of this podcast called This Week in Virology, which is like a bunch of really experienced virologists, just like Dr. Unnasch, pontificating about all the latest science. And so one of them recently said, in defense of the J&J vaccine, although the numbers in terms of efficacy seem a little bit lower, let's not forget that this is the right now vaccine. The mRNA vaccines were studied, those trials kicked off in July. So what we had in circulation then was more homogeneous, and less of a concern in terms of these these mutations in spike protein that affect how the virus acts and the selective advantage. And so this is more in real time. And the study sites included places like Brazil, included places like South Africa, but not, back in the summer, like recently. And it's more of a real world vaccine. So how it behaves in trials and the data that was used to provide authorization is what you see is what you get. And so actually, I think that if that were available to me, I would scoop it up in a second. I happen to already be finished because I work in a hospital, but certainly if for one of my family members that has not been vaccinated yet, and that's the one at the vaccine site that day, I would say, go for it. Absolutely go for it, because you will not die. That vaccine trial beautifully demonstrated that there were no deaths in the participant arms, the vaccine arm versus the placebo arm.
Yeah, and I'd like to point out on the other side of that, in terms of the safety of these vaccines, the latest study that I saw reported the results of the initial shots of 14 million people with the Pfizer and Moderna vaccines, and there were absolutely zero serious side effects that could be attributed to the vaccine. There were about 200 people that had an anaphylactic reaction to the polyethylene glycol that they use as the stabilizer in it, which is, a standard compound food item, and it's in a lot of thickeners and things. But for the vaccine itself, it was absolutely safe. I mean, there was no adverse effects that could be tied to the vaccine whatsoever. And another point I'd like to make with Priya is, the J&J vaccine has always been judged based upon only a single shot. The Moderna and the Pfizer's are an initial shot and a boost. So that also puts the J&J vaccine at a little bit of a disadvantage, which I think, based upon what you heard today, we're going to be getting boosters for these things for a while. And I imagine if you get a booster of the J&J, it's going to go up to 95%, just like the mRNA vaccines are. So absolutely. I agree. Whichever one you can grab first in your bloody little hand, grab it and go for it.
Right. And it's also nice to know that if you had some kind of concerning reaction, if you're in that small group that did anaphylaxis or had some other weird reaction to the mRNA vaccines, well, you have an alternative for next year. You have a vaccine that is recognized in an entirely different manner by the body. It's good to have multiple options.
So this is a general question about the life cycle of a virus. Does the most deadly variant become the most dominant variant at any point in time during a virus's life cycle?
That all depends. That's like the $64,000 question. There have been a lot of modeling studies that have been put out on that, and you can argue that there's pressures either way. The ideal thing for a virus would be to be totally asymptomatic and totally infectious at an asymptomatic level, because then people would never know they'd be infected and they'd be spreading this thing all around, and that would be great. The other side is like an Ebola type infection, where you develop incredibly high amounts of the virus in your circulation and you're super infectious and you die, and then your body's infectious for a while. And that's another strategy a virus might want to use to try and transmit itself. I personally think it's likely that this virus may have selective pressures on it to mutate, to become less virulent over time. And I think the reason for that is right now what we're seeing is a virus that's basically a wolf running around in a paddock of really stupid, naive sheep, right? Where almost everybody it comes in contact with is naive and has never seen the virus before. So the real selective pressure right now is to make itself ever more infectious and ever more efficient. The trouble with that is that the immune response is coming right against that spike protein, which is the receptor as well. And at some point, the virus is going to have to start mutating that spike protein to get around being destroyed by the immune system. And when it does that, I'm thinking we're going to start to see reductions in the avidity and functionality of that spike protein. It's going to be able to start to get around the immune system a little bit, but it's not going to be as efficient at binding to the cells. And therefore, it's going to start reducing its virulence, and it may end up just fading away as a common cold.
Great, thank you.
Can I ask a
Please,
follow-up
Dr.
question? We have an esteemed expert here. Dr. Unnasch, is that what happened with SARS-1? How did that end up just kind of fading away?
No, SARS-1 was actually the opposite thing. It was highly infectious, but only when people were really, really symptomatic. So it was really easy to identify that. It's just like Ebola. It's really easy to identify people who have Ebola, right? They're bleeding out Ebola orifices. So you could identify the people who are infectious, put them in isolation, and they could break the cycle of transmission really easy because it was really obvious who was sick. The trouble with this, of course, is that half of the people running around are not sick. They never even know they've had it.
Okay. So there was no a mutation or series of mutations that made it less fit over time, that it kind of just burned out?
Not that anyone's aware of. No, it was really easy to identify the people who were infected. And so they were able to isolate them really quickly and just kill it, basically.
So we have one participant who's asking the question. I understand the virus attaches to fat cells. Is there any evidence that the variants are more or less problematic for people with obesity or high BMI levels?
I'll turn that over to you, Priya.
I will say that I don't believe that this has specifically been looked at. I don't think that they've identified any risk groups that are particularly susceptible to these variants that are not already susceptible at baseline to any SARS-CoV-2 infection. So it's going to probably play out in the exact same way is what I'm assuming. We know that it has probably a higher attack rate, meaning that if one person in the household gets infected with a variant, a virus from the variant lineage, multiple people in the household are going to be infected. And if any of those individuals have high-risk conditions like diabetes or obesity, chances that they're going to end up in the hospital are already quite high. And it may be that once we've studied this for long enough and seen the impact of these variants, that they may end up doing worse. And that's what's been seen already it's been suggested with the UK variant that it actually may be more deadly. So if it's more deadly in the general population, then if you kind of put the lens on high-risk individuals, it's probably going to not be positive news.
Great, thank you. I think we covered this, but let me just, the questions come up several times, so let me reiterate the question. So we need to receive an annual COVID-19 shot the way we receive a flu shot.
Yep.
Okay.
I think that's my opinion, but yeah, I've been joking that our flu vaccine drives down from now on are going to be, instead of just roll up your right sleeve, it's going to be roll up your right sleeve and roll up your left sleeve. Get the COVID in the left and the flu in the right. Yeah, I think we do have a big advantage with this one, though, because the mutation rate on this is 30-fold less than it is for influenza, which means it's not nearly as quick on its feet as influenza is. And with the vaccines that we have now, we're much more quick on our feet and making modifications to the vaccine that can address any variants that come up. So I think we're going to have a lot less trouble staying ahead of this one than we do staying ahead of flu.
So is the mRNA technology and approach, is it applicable to other kinds of viruses?
This is the first time it's ever been tried on a widespread scale, but I'd say based upon what we've seen here, this is the wave of the future for RNA virus vaccinations.
So Dr. Nori, I'll give this one to you. If you've had COVID, do you need to be vaccinated?
Yes, Absolutely, you do. Because again, coming back to the topic of the day, the variants. The variants are making it look more likely that you can be reinfected with COVID. And I'll tell you, I've taken care of a number of people already who clearly had infection in March or April, were potentially even admitted to the hospital, had antibodies that we measured on our clinical test, and now nearly a year later, so like about 11 months, they're reinfected. They're potentially not as sick, but it is entirely possible. And so the best protection for someone who's never been infected or has been infected is getting that vaccine. Because studies show that some people get very robust antibody responses from the infection, but it seems that consistently the responses from vaccination even exceed that, and they potentially may be longer lasting as well. So yes, bottom line is yes.
Great. So we've talked about several of the vaccines that are out there. One participant would like to know the view regarding the AstraZeneca vaccine. Any comment?
You want to comment,
Yeah.
Priya?
So I think the concern is really with the South Africa data, that it really is not very effective in controlling virus new infections in that region due to the PY or the South African lineage that has been widely described there, and that is probably the predominating virus there. It doesn't hold up as well against that vaccine. And also there were issues with how, if you recall the data, initially there were funky doses that led to different doses given in different countries, and so that led to different degrees of immune protection. So I believe that they were asked to revisit that data before then presenting it again for emergency use authorization. I do feel like it will have its role somewhere, but we need a couple more months to understand exactly how that's going to contribute to the overall body of vaccine literature. Do you have thoughts on that, Dr. Unnasch?
Dr. Unnasch, anything to add?
Nope.
Okay. This will be our last question. So it's coming from a GIC member in the financial sphere, and from an investor's perspective, is the medical profession and industry entering a new era of innovation? And will medical advances for vaccines and otherwise benefit from this pandemic and the emergencies and the R&D that's taken place? Can you just say that from a longer term industry innovation standpoint? Dr. Unnasch, you want to take that first?
Well, I certainly think that what we've seen here, together with the development of a highly effective Ebola vaccine, which was based pretty similar onto the AstraZeneca type thing, we now have really advanced our capability in vaccine production. I used to always say that, you know, if we have an emerging infection, it's going to take 5 years, was the most quickly that another vaccine had ever been developed. So vaccination really wasn't going to help you with an emerging pandemic. But now we were down here to 11 months. My goodness. This is, you know, fantastic. I think the science has really taken a quantum leap forward. And I think it still remains to be seen in terms of developing new therapeutics against the virus, how that's going to work out. One concern that I have is that we may not learn that much about new advanced therapeutics because the vaccines are so effective. We may eliminate this as a public health problem, and the money is going to all dry up for the therapeutic research, and we may not get any new therapeutics out of this.
Interesting perspective. Dr. Nori?
I fully support what was just said. As someone who actually spends most of my time on therapeutics, The therapeutic story has actually been fairly, I don't want to say disappointing, but it's been a lot less exciting than the vaccine story. The vaccines are truly something that every scientist, everybody who's working in the sector can get behind. The therapeutics have been modestly beneficial at best if you take them in aggregate. We're always a step or two behind, or like 10 steps behind with the therapeutics, but with the vaccines, we have a chance to be a few steps ahead. Maybe now we're a few steps behind, but soon we're going to be neck and neck, and then even, I think, exceed the adaptations the virus is throwing at us. And so this is truly, in terms of biotechnology, since we've been tracking this whole story, sorry, I think there's some feedback, but the two really interesting aspects of the whole pandemic to me is, number one, by far, the vaccines, and the story behind testing, actually, and how quickly that developed, and how that capability in terms of public-private partnerships really ramped up very quickly as well. It still has a long way to go in the United States, but I think that's also a very interesting realm within the pandemic.
That's fantastic. Well, Priya and Thomas, thank you both for fascinating discussion and for spending time with the GIC community. We hope that you'll both join us again in the near future for another GIC program, but thank you very much. We certainly appreciate it.
Thanks a lot.
Thank you.
Thank you.
So audience, for our next executive briefing, it's going to be Tuesday, March 16th, and we will continue the examination and discussion around coronavirus and the variants for the coronavirus. So the registration for that event on March 16th is now and available on the GIC website. Finally, a friendly reminder about the matching gift campaign as part of the CoTalk Global Healthcare Series. Through the end of the first quarter, which is the end of this month, which is just a few weeks away, every dollar that's donated to the GIC will be matched dollar for dollar up to a total of $50,000, with all proceeds going towards the GIC's healthcare related programming. To donate or to register for next week's event or find a video of our replays, please visit our website. It's www.interdependence.org. As we conclude, we encourage you to take 60 seconds to respond to our audience survey. Your feedback is important to us, and we appreciate you letting us know how we can improve our programming. Thank you very much for your attention, and have a good evening.